Thank you everyone for joining today's AgBioData workshop. This is the last day of our community workshop. So today we will have presentations from an ending working group and 3 active working groups. So stay tuned. We have 4 talks to finish within an hour. So I would like to request everyone to mute yourself and let us go to listening mode during presentation. There will be 5 minutes of QA session after each talk. So if you have question, please feel free to write in chat box during presentation, or you can ask your question directly from speaker during QA session. So we will start our presentation with the data reuse working group, and James Koltes from Iowa State University is the speaker. And title of his talk is Perspectives on How to Improve Sequence Data Reuse from AgBioData's Data Reuse Working Group. So yeah, James, go ahead. Thank you.
Thank you. Thank you for the introduction. One technical point, can I simply begin sharing my screen?
Yes. We don't see your screen yet.
Yeah, one moment.
Yeah, you should be able to share the screen.
Yes.
Okay, can you see my slides?
Yes. Thank you.
Okay. Oops. So thank you very much for having me today. I think one of the challenges in talking about data reuse is there is so much that we could talk about that we could go on for much longer than 10 to 15 minutes. So I'll try to keep moving here. So we had a diverse working group, and in looking at what data is out there and what we can share, most folks realize it's not easy always to share data. There's a fair amount of work that goes into it. So hopefully this is not just something that we feel we have to do. Hopefully it's something worth doing well and getting good value out of it. And that's certainly the motivation behind our group. We have a number of individuals who are very actively reusing public data sets. Our focus of the problem was really related to sequence data because we were mostly interested in genomics and thought that was a good focus. We have lots of public sequence available at NCBI and the European EMBL archives. So that's really where we focused. The motivation for the data reuse working group really, oh, sorry. Thanks, Monica. I didn't realize we were on that display. Is that better?
Great. Yeah, thank you.
Thank you. Yeah. The motivation really for the working group was a fewfold. Again, I mentioned there are many sequence-based data sets. Obviously, we can obtain these data via the World Wide Web. There's an open science movement. There's more and more requirements to share data, obviously, and the federal government in the US is moving towards more mandates on that. But more than that, there's just incredible opportunities to learn about biology, and probably we're at the very tip of the iceberg, if you will, in terms of really using this data in applied ways to solve problems. There's just tons and tons of opportunities. Obviously, no data set is perfect. So, I mean, in reusing data, I think we need to think about every data set could have a use. When I teach about data reuse in my courses, one thing that we discuss is, a bad data set still has value if it's online, because somewhere you have to see an example of what maybe a non-ideal data set is. And with different experiments, different situations, sometimes data is what it is. That can create some challenges, but there are still opportunities there to reuse those data. Data are often underutilized due to insufficient information. And in my upcoming slides, you'll see me talk quite a bit about that, especially related to metadata that hinder the FAIR data standards. So as a group, really in talking about all of the different challenges that come with data reuse, we really focused on trying to identify bottlenecks and critical needs, and where possible, proposing solutions. With most of us focusing on agricultural genomics, although we did have some folks chime in periodically from other segments of genomics data usership. I will comment that as we tried to we tried to create solutions that we thought were something that could be done sooner than later. In other cases, I think we came up with more questions or needs, but I'll summarize those shortly. And then from a personal standpoint, I just wanted to mention my interest in data reuse is because I work on the development of databases. And in particular, I have interest in using public data to annotate genes, gene function, and regulatory regions. This is something that's kind of a hot area in my area of animal genomics. And so we're really looking at ways to link genotype to phenotype by taking public sequence data, identifying chromatin states, annotating what might be causal variants, and linking that to public data like the livestock QTL database and similar resources. So that just as a bit of overview to help you kind of see where our perspective comes from. I want to talk about some of the specific topics now that were addressed by our working group, some of the barriers, specifically data quality concerns, metadata and limited metadata in some cases was a second concern. Making data truly public when it says it's public is still a challenge. Interoperability of data, data ownership, in particular public versus private data, and how that can be shared. Considerations for data reuse for users with different skill levels, and then diversity, equity, inclusion in data reuse. So starting by talking about some of those specific barriers and then some recommendations to follow or some ideas to move forward in bold below these points. With data quality, our groups came to the conclusion that data standards are a solution. So data are made publicly available regardless of quality, or at least we hope that's the case, and then the users make a subjective decision on whether or not they can reuse that data. There's lots of factors to assess. I'm just putting a few of these out there, and if you've worked with genomics data, none of this is surprising. But this is a lot of information, and this is just a small subset of what's available. And so in part of that quality, complete metadata obviously is important. There's a limited scope of existing standards. I mean, one challenge with data reuse is that there's always this balance between getting people to share versus not. And there are rules in place, especially with funding to try to promote that. But that's a challenge because it does take time and investment and labor, and there's not always funds available from projects or as well allocated as we'd like from projects to get all of that information in. So it can be difficult at times to obtain information like experimental and computational protocols to help inform data reuse and meta-analyses. So our working group really proposed that there ought to be more ways to link protocols for data quality and related information, pipelines, and more statistical standards developed for reusing data to help the agricultural genomics field improve the use of data and handle data quality challenges in data reuse. Metadata obviously is a thread that flows through lots of the topics that I'm discussing today. And as we see it, we're hoping that maybe there's some way to incentivize more complete metadata. So much like the analogy of the iceberg, there's the data you see, and then there's all of the information that may or may not be there, often isn't there. Many times the metadata is incomplete, or we just have some subset of that information. When folks are submitting their genomics data, they're required to have a minimum set of standards many times, and there's a trade-off there because lenient submission systems obviously don't require as much metadata as possible. So we need some way to incentivize folks making good metadata available. In the working group, folks discussed, are there different ways to rate it, gold star data or things like this in some way? But the one true idea that came through is more need for citations, uses of DOIs, and getting this idea in the mainstream that citations of data are important. And probably part of this even permeates to the way that we look at tenure and promotion. As a young scientist who just went through that process, a lot of my colleagues don't always understand the importance of data and working with data, making it available and making it reusable, what that can do for the community. So another topic really is making data available to everyone when you say it's available, or what we call bridging the data availability gap. Survey from Science and Nature in 2021 found that an alarming less than 50% of data stated to be available upon request, but was actually not possible to be obtained, which is quite disturbing. And so really what our working group has proposed is that we need journals and particularly funding bodies to take a little bit more of an active role in making sure these data are available. The publishers certainly have gotten more involved in making sure that data is submitted to repositories, but there's still more room to go there. The federal government in the US, again, has this 2026 mandate that data needs to be public if it's publicly funded. And I don't know how that will impact our funding bodies, but right now, many of them have rules, but not very good ways to enforce those rules. And so sometimes we see really nice data sets that are generated, but they're never actually publicly available because it's not possible to really enforce that data gets out there. Oops. Another challenge is interoperability, and we think this comes back more so to data formatting in some ways. Our community has converged on a whole subset of different standards, be it FASTQ, SAM-BAM, VCF, GFF3, BED, and so on. So we don't think the issue per se is in standardization. We've got formats that have good standardization, but perhaps consistency of use in some cases, or the ability to work between these different formats. Sometimes different reference genomes and reference genome mappings can be an issue with annotations. I know in my community, we have challenges with this because we're constantly generating new genomes, or now pangenomes are coming like crazy. And the question is, how do you quickly and efficiently compare information across genomes? So there's need for some consistency there and probably good communication as well. Developing ways to be backward compatible, to take outdated or older genomes or lab sequencing methodologies to be combined through extensive metadata with new information is one thing that might be helpful. And then also, I think rapid and efficient coordinated ways to compare annotation sets that may be aligned to one genome or one data set. If it's important, having ways to rapidly be able to align that to other genomes, especially again in light of pangenomes that are coming on so rapidly is really important. Then the topic of data ownership, which always seems to be looming, and how we share data. So having access to relevant, affordable study populations from breeding companies can certainly be very helpful. The challenge there obviously gets to be the intellectual property piece. If data are generated through public funding, then there are already lots of sharing requirements. And this upcoming 2026 mandate to make data available is obviously an important one. But we still have a need to continue to develop computational approaches to protect industry data while allowing it to be used for research. My sense is I'm not sure if this is as important in all communities, but in some communities, this can be the difference between having access to readily applicable data to the industry and not. Another challenge I think that our working group felt was maybe at times underappreciated was thinking about user skill level and resource availability. A lot of times when we talk about data reuse, we talk about getting sequence data, for example, out there. It's at the database, great. But do people really know how to access that, retrieve it, use it, check it for quality, go through all of the processes that are needed to use that data in an effective way? And so a recent study showed that, the skills or perceived ability was identified by many participants in reusing data as a major factor influencing their behavior. And most scientists in this 2017-2018 survey from Phenotypic were either highly or kind of mediocre in their risk averseness to reusing data. So there's some need to kind of help ease some fears and make people more comfortable, probably in part through training. In the US, this is likely not an issue due to lack of computation. It's more of an issue of need for training and improving folks' skill sets at various levels. So there's a need for education programs for awareness raising and developing good practices in data reuse and incentives. And so this DataWorks prize is pretty neat. If you check out that link, you can actually win awards for coming up with clever and innovative ways to use public data to do cool science. So finally, moving kind of into the last stretch of this talk, future of data reuse, the importance and benefits of inclusions and some takeaways. Obviously, to have fair data and to be able to share data well, we need to make sure that everybody can reuse this data. There are lots of benefits from data reuse. Having diversity and lots of different folks at different skill sets, different talents in different fields will help breed innovation. We need to have all of the abilities, resources available for folks to be able to do that, be it computation, digital literacy, et cetera. And we need to be respective of different cultures. So a couple of takeaways, barriers and limitations that we've discussed are listed here, but there are many opportunities in reusing data. Looking ahead, I mean, the opportunities to use AI and machine learning with these big data sets are huge gaps and opportunities. There's many other data sets that could be used, phenomes, microbiomes, all of these different omes. I think these are opportunities potentially for a new working group. If I were going to give advice on where to start, maybe with sensing or microbiome data. Deliverables, our group has generated a white paper, and you can get that from this QR code. This is my last slide, except my question slide. So I think I'll stop there since I know I ran just about to the end of time. If you'd like to grab a shot of this picture. And I really need to acknowledge Alenkna Hafner, who really helped with many of these slides and has been a champion in pushing forward our white paper, helping us get it out there, getting it submitted. So that it's available to everyone today. So thank you for your attention. I don't know if we have time for a quick question.
Thanks, James, for the presentation. I can, if someone has a quick question, I can please ask one question. I can give you time for only one question. Okay, so let us move ahead. And the next speaker is Jinha Jung from Purdue University. And he's from Phenotypic Data Standardization and Management Working Group. And title is Furthering the Phenotyping Data Ecosystem for Genetic Genomic Discovery. Yes, Jinha, please go ahead. Thank you.
Thank you very much, Sunita. I hope you can hear me okay. And
yes.
I'd like to give a quick update on newly created in a working group, which is in a phenotyping data standardization and the management working group. And this working group is co-chaired by Jennifer Clark. She is in this room, I believe. And we initially start with Jennifer and I agreed to co-chair this in a working group, but we soon realized that there's a lot of people interested in this. It's not going to be a 2-people job, so we invited 2 additional key contributors to this working group, which is Peter Shelby. I believe he's in this room as well, and Jennifer Lahowick. And she is, I believe, not in this room because she's on travel. Hopefully she can. But I'd like to acknowledge that those are the key contributors to this working group. So, as I mentioned earlier, this is a new working group that we just started earlier this year. And when thanks Anarita, she had some kind of survey to gauge how many of those members in this AgBioData the consortium are interested in this newly created working group, and the response was immense. I think I don't remember exact number, but I think over 40 responses that they are interested in joining this group. So when we were planning to have a kickoff meeting, we thought that we need to have some kind of pre-kickoff meeting survey to be sent out so that we figure out when is the best time to meet and how many are actually willing to join, because even though there's a lot of times you said I'm interested, when you open it up, we never know that how many is going to actually show up. So we had the kickoff meeting survey, sent out. And we asked, three questions. The first question is what time zone you are in. And as you can see, I think that there's most of them are, the US based, but you see the GMT -5, -3, and etc. So we realized that there is the members who is interested in this group is from all over the world. So it's going to be very hard to find a time that meet everybody's needs. So I think based on this survey, what we decided is that we're gonna meet every Tuesday, 11 a.m. Eastern time. That is gonna be not too early for majority of the members in United States, because 11 a.m. Eastern time is going to be, I believe, 8 a.m. and the Pacific time. Plus, that's going to be also not too late for European members, but the only downside is that that's going to be very late at night for the Asian the members. But we thought that we cannot find the time that is going to meet everybody's needs, so we just decided to go with this 11 a.m. option. And the second question that we asked was, which subgroup we are interested in. Initially, we are thinking about having 3 subgroups because we had a pretty large number of members interested in joining this working group. So we came up with these 3 subgroups at the beginning, which is phenotyping data standardization, phenotyping data generation, and the phenotyping data management. And just okay, I was second the chat. So based on this response, 50% of the response said that I'm interested in data management. 38% is data standardization, and 11.5% is data generation. Based on this, we thought that maybe we have a little bit too the pie for the data generation is a bit too small, and data standardization and the management is quite different. So probably we can merge data generation and management into one, and later we decide to only create a 2 subgroup based on this response. And we also need to set up the kickoff meeting time, and we can send out the survey to find the best date, but we just went ahead and proposing these 2 dates that we're gonna meet, because we have a large number of people. I thought that doing a doodle poll is not gonna make sense because there's gonna too many variation in terms of preference in what time and what date that we'd like to meet. So we went ahead with the more top-down approach where these are the 2 dates that we're gonna meet. Hope you can join this, and let us know whether when you can join, or let us know if you cannot join at all. And it turns out that more than 95% of people that responded to the survey saying that they can either join the February 22nd and 27th. So we did that. And we had 2 kickoff meetings hosted in February 22nd and 27th, 2024. And this was mainly about getting to know all those members who are interested in this. And we have started with this kickoff meeting with self-introduction. And these are the description that we initially developed when we sent out survey to gauge who might be interested in. So I'm going to go through this in a description so that everybody understands what this working group is about. And that this working group will address the need for standard guidelines and expertise for phenotypic data management that can be implemented by the consortium member database and the large community of data users and generator. In particular, this working group will identify and provide recommendation for 2 categories in the phenotypic data usually created by the members of DB resources. First one is the previous the data set is available from literature. The second one is phenotypes submitted directly by the data generator. And like I mentioned earlier, based on this, we decided to create 2 subgroups. First one is phenotyping data standardization, and this subgroup will be led by Jennifer Clark and Peter Shelby. And the second subgroup we decided to create is phenotyping data generation and management. I think the logic behind merging those two into one is based on the pre-kickoff meeting survey, and myself and Jennifer Lachowicz agreed to take the lead on this in a subgroup. And these are the key deliverables that we like to make by end of this year one, because we only have year one, and this is going to be for only 1 year. And this working group is a part of this AgBioData consortium. So whatever the discussion or things that we're going to deliver should be aligned with the interests of AgBioData theme as well. And the one thing that we made it clear at the beginning of during the kickoff meeting is that this working group is going to be about learning how to process your data. This is more about learning about what are the the phenotypic data standards out there, what are the best practices for generating and also managing your phenotypic data. And we like to develop a white paper at the end of year one. That is something that we'd like to achieve. And we also like to provide a recommendation to our community, and based on existing or some of the needed best practices and tools. And after this kickoff meeting, since March was kind of crazy month for both Jennifer and myself, so we decided to skip the March one and decided to have our first meeting in April. So we had our first subgroup meeting in April 9th for data standardization, and data generation management subgroup met on April 16th of 2024. And we only had a first meeting, so there's nothing too much to report. But in the first month subgroup meeting, we mainly focused on to develop vision statement and what will be the specific task that we like to tackle for the following years. I believe this is the last slide, and I'll be happy to answer any question you might have from the audience.
Thanks, Jinha, for the presentation. Yes, we have time for one quick question. If someone is interested, please raise your hand or ask directly. Okay, so I don't see any question. Okay, thank you. Thank you, Jinha. It was interesting. So let us move forward. And our next speaker is John McNamara from Washington State University, from Diversity, Equity, and Inclusion Recruiting Working Group. And title of his talk is, How do we address all the aspects of diversity, equity, and inclusion in AgBioData? John. John is there? Yes, I see John over there. Yes, John, please start.
John, I will write him.
No, sorry. I had a computer problem right there. Okay. Bear with me a minute. Sorry about that.
It's okay.
Can you share the screen?
Yep, I'm getting there again. Sorry.
Do you want me to share the screen for you?
No, I've got it. You see it there?
Yes, we do.
Thank you.
All right. Sorry about the delay. Okay, good morning. Thank you. So the Diversity, Equity, and Inclusion group merged with the recruiting group during the project. These have been the members here lately, and thank you for that. The inclusion of DEI was part and parcel of the original RCN grant to expand the network to all stakeholders. And a little bit differently, and so not only diverse groups, but groups of different researchers, different databases, those types of things. So really, an all-out effort to expand this to everyone working and interested in ag biodatabases. So already we have a number of different folks on the group, from libraries, from private organizations, from institutes of higher education, federal agencies. So that we have been successful in that. And in a minute, I'll reach out, or I'll talk about where we reached out in terms of the 1890s and tribal schools as well. Okay. And so we've had a number of different seminars. We had invited Dr. Rose Marks and Alton Thompson and the group from the Agricultural Research Service, the ARD group of the 1890s and tribal schools. And we met with them almost a year ago, I think, 7 or 8 months ago, to discuss some of the things that they're interested in. And there's one next week from SACNES, Dr. Charlotte Lambert, and I saw on the emails this morning that that's been organized. And so she's going to talk to us about their efforts. And then this past January, after we made a number of different discussions, we got on the agenda for the ARD business group. And because it was a big meeting for them, going through all of their issues and management, it was just a very short introduction that I gave on what we're doing and some of the things that we might be interested in collaborating together, as has been brought up by the previous two speakers as well. So, and thank you for Anna Rita put this in. How, and part of the discussion today, here in a few minutes, I would hope that you have different ideas. If you have specific working relationships with folks that you want to introduce, or some of the things that we can talk about. And I want to use this opportunity, several of us has brought up in the group, is that one of the things we want to avoid is coming in, hey, we've got this, would you like to do it, type of thing, but really to listen to other people to say, what are the things that you need? What would be the things that make your life easier and more effective? So please be thinking about that during the discussion and beyond. So this is the last slide. And really at this point, we are, the ball is in our court, so to speak. One of the things that we always have to be careful about, is that, okay, we've made contact, we've ticked that box, but we really need to, at this point, where are we going? And I made it the comments in some of the meetings that we've had over the last couple of months, is that, and we've discussed ending this committee or this working group with the charge that this is now part and parcel of what we do. So all the different activities that are going on, we will reach out to underrepresented groups and see what they need. And so the next follow-up, and they've said, go ahead and do this, call us when you're ready, with a couple of specific members of that group to talk about, okay, what would be the best ways forward? And the final thing there, I put this link in and I'll put it in the chat. At this moment, the National Academy Board on Agriculture and Natural Resources is following up on the land grant study that we did 2 years ago, almost 2 years ago. And this is specifically, this impactful collaborative platforms is specifically to study and make recommendations on how we can be inclusive of all the universities, historically Black, tribal schools, underrepresented schools. So I'll put that in the chat later. It's something that you can provide feedback for. And whether it's through AgBioData or your own work. And acknowledgments, and I really want to call out Anna Rita for pulling this together. She's been steadfastly working to keep this group going. So with that, I'll turn it back over to the moderator.
Thank you, John. Okay, any questions from audience? We have time.
I gave you a few minutes back.
Yeah, thank you.
I have a quick question, John. It's more about like how to best reach out to the 1890s, the HBCUs, right? It's never been clear to me whether there's a contact in USDA that facilitates like communication with the 1890s and the HBCUs, because as land grants, we're always, being encouraged to reach out to them. And I'm always like super supportive of reaching out to them. It's just the question of like, how,
Right.
how to
Right.
do that in a way that's effective. And I've never gotten a good answer on like how. So I've always, been like, hey, you colleague, I happen to know who's at one of these. Like, could you, what about this opportunity? But it's always like the one-offs. And I'm constantly trying to figure out, okay, how do we do this in a way that scales? So
Right.
it's not like, and it might be, it's not me reaching out usually to say, hey, I have this thing that I'd like you to do, or this might be interesting, please do something. It's more like, here's an opportunity that we have resources, and if you're interested, like just to advertise for us, we're happy to engage and do a lot of the work for you. But I've only ever had like one-offs, where,
Right.
and I'm always, I just don't have a good sense for, if it's like land grants, I can kind of go to APLU, right, and try to get the message across the land grants, but I don't know if there's a analogous
Sure.
organization for
Well,
them.
I'll give you what I know and what I've done on that. So yes, there is that office in USDA. Don't ask me what their name is right now. I can't remember, but we kind of turn that around, if you follow me, that not us, like, okay, there's a grant call. One of the tick boxes is, get underrepresented people involved. You reach out. Okay, turn that around. And that's why we initiated the conversation with their management group. So their consortium of ag directors, okay, is what that was. What are you doing? And I know from following it is that in the last 2 years, that population writ large has been, I'm trying to get the right adjective, provided with massive, massive amounts of money for research and education. So really the other part is, what are they doing that we can help them with?
Yeah. Okay. That's great. Thank you.
Yeah, and in the chat, yeah, Susan has been a contact there in USDA. Thank you, Anna Rita.
Yeah, great. Thank you.
Thanks, John, and thanks, Jennifer, for the question. So our last speaker before breakout session is Karen Yuk from Caltech. She will provide updates on FAIR Scientific Literature Working Group today. So please go ahead, Karen. Thanks.
Hello, everyone. So I'll be reporting from the FAIR Scientific Literature Working Group. This is our, let's see, our update. These are the members. I don't think much has changed. We haven't gotten new members that I've met yet. But what we've, sorry. Okay, so sorry. So we started with trying to identify bottlenecks in the publication curation pipeline. And we reached out to people and discovered that. We identified some tools that we could do to help with that. And I'm going to explain that to you. And then, our final outcome is going to be a published recommendation for roadmap. So when we looked at what stakeholders, what the challenges and barriers were for stakeholders in getting their data to be fair, they came up with a number of different, divergent but also similar problems. And the one problem that we thought we could really address was this one systemic problem, which is not knowing where data should go. So we focused on that, and we tried to figure out what would be the best way to overcome that barrier for people. That we all kind of know that data should go into a community database that's open and accessible to people. But, one of the problems, of course, is that there might be more than one database for a community. So this would be a bit confusing, such as wheat data could be found in the Triticale toolbox, grain genes, and Gramine. The other thing is that all databases not host the same data types. So depending on which database you go to either find data or to try to submit your data, you really need to know which one to contact. And then some databases only allow data submission from individuals. So not all databases will take submissions. So having people direct you to a database that does house data that you create isn't really the appropriate one for you to go to submit, such as Gramine, who takes data from other databases. And then, of course, there's a quintessential problem that there may not be a database for the community. So how do we address that? So one of the things we did was to just realize, okay, how can we create something, or how can we help people find the appropriate database to put their data in? And we came up with, well, let's make a tool. First, to get that together, we needed to understand how, using the AgBioData Consortium as our playground and as the group we're serving, we wanted to understand who the database members were and what their crop and data focus is. And the other thing we had to do was to figure out what type of data each database maintains. As you know, everybody in their own communities uses their own terms. So one thing that was really important is to make sure that we were all talking about the same type of things. In order to do that, we mapped terms, all the data type terms that all the databases use, and we matched them to mesh terms or EDM ontology terms. EDM is an ontology for conceptual biology and bioinformatics. So I think going forward, this is going to be an important thing to do. The next thing is we need to figure out if the databases allow community submission or if they only take from certain repositories. So this was quite a bit of work to go through, because we do have a lot of members, and the member databases do have a lot of different data. So what about data that should go into non-community data repositories? So we all are familiar with NCBI, INA, and EVA, and these take specific data and large data sets as well, and they are the source for or are the source repositories for other community databases. So especially, NCBI deals with the sequences and SRAs, and EVA, we had to talk about that earlier. It's very specific for variations. And we want to encourage people to go into deposit their data there when appropriate. Okay, so what about data with no community database? We're still working on that, but I think one of the main things is just alert people, about their options when they have that type of data. And of course, the big question is, well, is there already a tool that does this trick? And the answer is yes. So at this point, I'm going to I'm going to do a new screen share. Where is my, sorry, let me get out of this. Shoot. Stop share. Oh dear, sorry. I have to get to I think I just lost my talk. Sorry.
It's okay.
Okay. Okay, now I'm really confused.
So,
it's the curse of the live demo.
Well, yeah, and being sick and not being able to really work on this for the last day too. Okay. Just gonna share my desktop so I don't. Just back up here. Okay, so this is a tool we came up with, and I think some of you, we tried to get some feedback from the group before, and we thank the people who did give us feedback. We haven't changed anything since you given us that feedback. So the issues that
Carol,
you brought
I
up.
don't think we are seeing your the right screen.
So we are not able to see your presentation. Yeah.
Now
There.
it's good.
Yes.
Thank you.
Now we can see that. Yeah. Thank you.
Okay. So like I was saying, the issues that people had given feedback for before, they're still here. So you're welcome to remind us what those are, although we have it written down. So this is a tool that was built by Cornell, and I'm blanking on what it was originally used for, but it's a tool it's a resource finder. And it's made in Drupal. This is really the work of Suk and Catherine. It's an amazing amount of work because Drupal is not so easy about just loading things in, apparently. So as I mentioned, we had to figure out all the different databases that we have. We categorize them by organism. This is only a snapshot. This is a prototype. There's a lot more development that needs to happen on this to get it complete. And I can talk a little about what the vision that we have for the AgBioData Consortium, but we want all the databases listed here. We also had to go through, and this was the important part of mapping, their terms to standard vocabulary so that we can easily sort. So the way you use this is to just, we envision people coming, they have, I have cotton data. And so they have cotton data, and then they will find which database has their data, and they can look more closely at what the database is about. And we made short descriptions for everybody, and also a list of the data types, that they house. We also will need to have, a separate section for the data types that they allow submissions for, or where they take the data type from, so people will know. We give the submission page, so people easily can go there and look up what the database has for that, and contact page, etc. So for cotton, in this case, it looks pretty easy. But if you have, let's see, yeah, so it's pretty easy to find right now because there's only one. But if you come and you don't know what you come and you can look through by data type. So if you have genetic map data, just by selecting it, you find all the databases that do take genetic map data, and you can pick those and really do a comparison about those to find out, is this really the database that we want? So this is really useful, especially for places that, well, they might not take the data, but if you look through, you can find that, they house the data, but they might not take it. Okay, So we do want feedback on here. So there are things that don't work because of the software. So, and it's not easy for us to deal with. So this is, as I said, a prototype, and we hope you like it. It's been a lot of work. Okay, so let me go back to my presentation. Okay, so did I do that? Okay. So what we hope is that this would live on the AgBioData consortium site. It can also, you know, the member databases can also might have this. As I mentioned, it's going to need developer support. This is a great prototype, and I hope you agree. We welcome your feedback, and we'll have you, you know, have a chance to play with it in the breakout sections or sessions. There certainly are more features that are needed. We'll need feedback for that, and like what's missing and what else might not be needed in there. Okay, so this tool, I think, encompasses, you know, some of the major problems with, you know, major roadblocks of people getting their data into databases. You know, it'll be able to tell you which database, you know, covers your field, what database accepts your data, and you know, what databases don't. So we think we've kind of addressed this one challenge. So the next thing is, well, what else can we do? I think one thing is it would be nice if all the databases had a similar tool, not just about you know, all the other databases, but within their own database, what they do. And, you know, like when they come in, you know, they can say, you know, I have this data, and the database can also, you know, reinforce, yes, we take this, no, we don't. This might need to go to NCBI, this might need to go to, you know, so those need to also be on that, on our main site, to help people find out. Also, just building more modules for how to submit data, so we can, you know, work with the databases to, you know, just keep, you know, just even the submission language the same, so that people know from place to place what's going on. And then the other thing is, you know, not all databases are fair. So one thing, you know, as we are the Fair Literature Working Group, it'd be good if we, you know, just reinforce the fair practices for data management and data housing. Maze GDB has a very good, I'm putting a little link to their page of fair practices. I think it summarizes things for databases really well. So we can, you know, try to get that out to more databases. Okay, so for our breakout, sorry, this is an old slide. There's something on there that shouldn't be, but we're going to allow people to play with the tool, and then we have some questions. Okay, so that's all.
Thanks, Karen. Thanks so much. And I really like this database finder tool. I was also beta tester of this tool. Yeah, so wondering if anyone has any quick question from Karen.
Monica?
I have a quick question. Thanks, Karen. This is cool to see. And I was wondering, from your perspective as a publisher, I mean, one of the use cases for this tool would be, you know, I'm a scientist and I'm currently generating data, or I'm finding data, or I need to find data in the agricultural space. How do I get that? But another use case, I imagine, especially from a publisher perspective, is we have reviewers who need to ensure that the data that is in the publication that's being reviewed is in the correct place. So have you thought about, as a publisher, you know, using the tool in that scenario as well for reviewers?
Absolutely. I mean, we have two publishers actually on the Fair Science Literature Group. We have people from genetics, GSA, which publishes genetics and G3 journals. And then, of course, I'm part of micropublication along with Daniela Ricciuti. We are constantly thinking in terms, you know, from the perspective of the publisher. And I think, you know, we work very closely with the databases to find a curator from each database to help vet, you know, the articles, the data that comes in. So, this tool is very cool for us because we can look and say, oh, okay, we have a wheat paper. Where do we go to find a curator to help us make sure that whatever they're reporting meets community standards, nomenclature, and we can guide them on that. Absolutely. Thank you for bringing that up, Monica. Peter, do we have no more time? Sorry.
Can we ask questions can we discuss in during breakout session, because we really do not have time now?
I think we have time for one question. So we don't have a break today.
Yeah. Okay, go ahead. Please feel free. Yes.
Peter?
Peter?
I guess not.
I have a question. Can I ask? This is Sushma.
Sure.
Yeah, so I think it is important to differentiate between the primary data repository and the secondary knowledge bases. So secondary knowledge bases don't take data directly, as you mentioned, but they synthesize the knowledge, and also they have to integrate many different types of the data together. And then the other part is that you said that there is no consistency in terms of the tools and the features different databases have. And I think it is a good thing because they can offer a variety of things, and the resources are not duplicated. So idea could be that they could connect to each other for the features and tools and analysis and data they offer. But at the same time, if every database becomes the similar way, then it will be lots of duplication.
Very, very good points. Yeah, no, the point is not to disparage any database that's part of the consortium, and really just alert people to how the databases handle the data and take the data in. And of course, every community has their own, speciality and needs for their community. So they have to service their communities, and those are not going to be the same across all databases. So absolutely, I am a full proponent of having lots of databases and not having just one massive one. We're kind of dealing with that right now with clients, but no, absolutely. And this is actually a really good part of the tool, because we can highlight, the differences, and, where people might not be able to put data in one, we'll be able to highlight that this is a good database to put your data in, because they can handle it. And I think it'll be really important to find out what data doesn't fit in any database, so that it can be, somebody might be able to figure out how to adopt it. So yeah, those differences are very important. Thank you for pointing that out.
So in the white paper that we had put together for genotype phenotype, there is a big supplementary table that has a list of all the databases and what they do, what type of data they take, and which are the primary repositories, which are the different knowledge bases that are geared towards a specific aspect, like pathways, like transcriptome, like ecological type of data, and so on. So maybe it may be a good idea for you to have a look on this supplementary table.
Well, that was a G2P.
Yeah,
we use that. Sorry, I didn't give you credit for that. No, that was incredibly important in the beginning. We took that and we used it. So you saw the amount of data that we had to, or amount of information that we went through, and we categorized, and, we did have to put things into subcategories. So yes, thank you very much for that. And I'm very sorry I didn't call out. I wasn't sure who the person was, but yeah, we use that. So thank you.
Thanks, Karen. So now we will have a parallel breakout session on 3 active working groups, and I will.
So this is the section where we describe what we discussed in the breakout session. So I believe we have about 15 minutes and 4 groups. So we have maybe around 3 minutes each.
There are 3 groups.
There's 3 groups? Okay.
Yeah, 5 minutes each, more or less.
Okay, so we got about 5 minutes. Okay. So yeah, so the first would be the phenotypic data standards group. Whoever was a scribe could go.
Oh, okay, Carson, I can talk. I didn't have time to make slides. I was too busy writing in the Word doc. So I'll do a 5-minute hand wave while, as in replace of slides. So we had a great conversation, and I'm happy to have people step in if I forget to mention something. So we talked about 3 different things. We talked about what should be the objectives of the working group. We talked about what are the outcomes that the community would like to see. And then we talked about how we're going to measure success. So under the objectives, we initially heard from Sushma, and she gave us really nice background and overview of what the phenotyping and genotyping working group had done, and they had made a lot of progress. They had an excellent white paper that does include a lot of information about phenotyping data. And we talked about, a good objective would be just raising awareness among the community of like what already exists, right? The white paper had a nice list of phenotyping databases. I think there's more of that out there. So just raising awareness of, okay, where's the phenotyping data that we currently have? How can we access it? Where is it all located? Is one. 2 would be trying to get a handle on what are the current practices for managing phenotypic data? What are people in the community already doing? Are there things that we could use to put together some starting best practices around phenotyping data? And then, one of the outcomes I think that we'd like to see is, answers to questions such as, I'm running a genetic database. I don't really, but I'm making this up. I'm pretending I'm running a genetic database in a certain agricultural species. I would really like to link the genetic information that we have to phenotyping data, because that's part of addressing scientific questions. It will enable the community. What is the best way to do that, right? How would you go about doing that if you wanted to link the phenotyping data that's relevant to your own database? We heard about Peter told us about there's standards out there, minimal standards for phenotyping data. There's some ISOTAB standards. So, finding that information and sharing it with the community, just kind of make like a map and scope of what exists would be super useful. Then we talked a little bit about how challenging it is to work with existing data that doesn't necessarily have good ontology associated with it, or it's very confusing. What exactly are the traits? And so the sooner we can kind of bring the community together to adopt some standard practices around how to label phenotypes, and then how to get some consistency would be really super helpful. And let me see. We mentioned some existing groups that are working in some of the phenotyping spaces. One of the challenges that we have is that there's so many different phenotyping modalities and different sensor types that it can get challenging very quickly. But there's some spaces like with UAVs where members of the community are starting to come together and set up best practices. So we wanted to make sure we highlight that. What we'd really like to see at the end is a white paper that summarizes like what is going on in phenotyping data, what are some of the existing tools and solutions that are out there that would contribute to best practices, and advocate with USDA to and federal agencies on how important it is to provide some resources so that we can, so that our existing genetic databases can make the most out of the phenotyping data that's being collected. Okay, if anybody else wants to add stuff, please do.
Thank you, Jennifer. I think you did a great job. Nothing to add from my end.
Okay, thank you, Jennifer.
Questions? Yeah, if anybody has questions.
Okay, if not, we can always circle back to you, Jennifer.
Thanks,
The
Carson.
next is the DEI and Recruiting Working Group.
Hey there. So we had a small and powerful group, myself, Monica, Josh, and Sarah Dyer from the UK, and Stephen Slota from University of Texas. And I'll just summarize on, we had some really good discussions about how to make inclusion work, how to embed it in everything that we do. And in terms of the RCN, a couple of things to then consider is, all right, so we've had this working group, we've made some contacts, we've discussed a lot of potentials, but as I said in the summary, okay, now is the time for all of the working groups to be specific and explicit in how they're going to pursue inclusivity and equity in those areas. And also, as part of the goal of the RCN and with our extension, we can then have specific goals moving forward. So what would be some sustainable programs after the RCN is complete? And then that really falls into the member databases. How can the member databases work within themselves and together to be inclusive? And a couple of the things, as we've talked about, for those of you that have been on the different working groups and steering committee, we've talked about it. For the rest of us, is okay, the education modules, the education course that has been put together is an outstanding possibility. And that conversation really needs to take place to say, hey, we have these modules on database management. Do you think your students would be interested in that type of thing? The other thing, more specific to the United States and kind of the traditional ag mission, the HBCUs, the tribal schools, there is a large number of funded programs from USDA and from the White House on data use writ large in underrepresented schools and communities. And so for those of us that have been kind of hardcore genomic, phenomic researchers and have had that career, and that's what we're kind of focusing on, the details of that, I think, we talked about, okay, expanding that out. What are some other users? What are some other funding sources that may not as be concerned about the details of metadata, but how can we use database management to solve problems in the field or make more efficient climate-friendly types of work? So that I think sums it up. Higher level summation. We had a great conversation, and we'll be following up with the steering committee. Be happy to answer any questions if there's time.
Okay. If there's no questions, we'll move to our last working group, which is the Fair Scientific Literature Working Group.
Hi, I, Jacqueline Campbell, will be reporting on that. Sharing my screen so you can see the slides. Okay. So the first question basically was, what else can we do to add to this tool? So should we add other databases like non-community databases? and what should display and what will they accept. I mean, this could be part of it, like for example, NCBI, EBI, and other ones, just to have it there. I think that's something that the group itself still needs to kind of think about. What can the individual databases do with this tool? Since it is a Drupal tool, and not all databases are Drupal, of course. The best thing that I think, and a lot of people agreed, is basically just have a link for it. So maybe, encourage other databases to link to this tool once it's complete, and even make a template of the based off the work that Carson's group did at MageDB about their fair practices webpage, for others to kind of look at, and of course give MageDB credit for all the hard work. That, I bet that was Lisa's work more than anything else. And then the Drupal tool requires a lot of data input. And, it's only done once, but like once we do it, we need to do it occasionally. But maybe having a developer to assist to make modifications to the tool if we had developer time, because it's basically a curation issue, inputting all that data. Other things we can add to it is add acknowledgments to the page, where we got the original tool from the Cornell University Research Managing Service, and also give the G2P working group credit for the really nice submission table, supplemental table that was published. One thing also brought up was that this is really a discovery tool for users, not really a data submission tool. So like maybe the idea is for like if individual databases want to make like a data submission type of tool to go into more depth about the specifics about how to submit data to their databases, to the right submission form, or a validator they might want to use. There you go. And then what can we do next, like after the tool? We talked a lot about publications with no data available or claim that data is submitted and it really isn't there. A couple of examples came up, especially during like the last working group 2 days ago. Was like there was a file project number in the publication and it's under embargo, so you have to contact NCBI and tell them all these things. There was even an example of a fake bio project, like it wasn't even there. Another example was like the bio project was there, but it was draft, and so it was just they submitted it, but there was errors with the bio project submission, and they couldn't release it. Another question really was how to engage publishers and find funders to really use this tool. And that was a lot of discussion about that, but like if we require publishers to have a submission along with a paper, then the database needs to be able to, manage this and have a review space. So like really seeing the data when it's being submitted to the journal. There was a discussion about how like NCBI could have more info on embargo bio projects. Like if you look up the bio project and it just says, oh, not here, it could say this is under embargo. So then we can at least know. Or even there was talk about even having an AI checker where once the paper is published and it has that bio project in the publication, that really the data should just be automatically made public. And then Karen mentioned that she will be speaking at the Society of Science Editors meeting and actually will be discussing some of the issues that database curators brought up. One specifically that was brought up by the group, the breakout group, was like request journals to be a little bit more firm in their data availability statements, like saying like things just if it could be available. Oh, the Council of Science. Oh, Council of Science Editors. Okay, sorry. But just trying to talk to more people about our plight and how we're trying to help society or science community as a whole. So that is all I have.
Okay, thank you, Jacqueline. Any questions or other feedback? Okay, if not, I think that wraps up the breakout recap session.
With this, we conclude the part related with the working groups. And now there will be a 30-minute session where we will talk about the future direction. We will talk and discuss with you the future of AgBioData. And this section will be led by Linear, which she will start with a presentation. Right, Linear?
Yeah. Remember how to share a screen. All right, I think it's this one. Oops, you got it?
Looks good.
Okay. All right, so yeah, I think I'm giving the talk and maybe other people are helping to sort of go ahead and moderate this. So it's my pleasure to introduce this terminal section of our workshop and give you some thoughts, concluding remarks and thoughts about the future. So first off, I want to thank and acknowledge all of the people who participated in all of this work, for the current workshop, as well as all of the work we've been doing for the last couple of years. So thank you all to the speakers and moderators, reporters and scribes, for Anna Rita for making this all magic work so seamlessly. I really want to express gratitude for all of the members of the working groups who have put in and dedicated so much time, the members of the steering committee, the external advisory board, and of course to acknowledge the support from the National Science Foundation, without which we would not be here today. So thank you very much. So, the sort of summary is like, oh God, we've done a lot of work. This is just amazing. I have enjoyed so thoroughly hearing all of these talks and reflecting on all of our previous workshops and all of these accomplishments. Like, we were so ambitious. The original proposal was so ambitious. And then we crammed 4 years into 3 years, and it's just been amazing. So there's been so much work done to identify challenges, to make some recommendations, to find the gaps, and have some really hopeful forward thinking about what we can do in the future to help everyone make better use of data. And I think there's been a lot of ideas floated for collaborations and tools and resources that we need. This has been a lot of hard work and a lot of time that has been gifted to us. And we've learned and are still learning a lot about how to build and sustain this community going forward. And so we've really, as noted, have grown our network, and we've really increased our capacity, and that's been very gratifying. So I wanted to sort of frame this kind of future discussion by thinking about like some of the things that have changed since 2020, when this grant was first funded. And people have mentioned and alluded to this before, but we have in particular, executive branch directives regarding the future of data handling, about the open data science requirements. We have the Biden administration support for fair data, and in support of advancing the bioeconomy. And I was privileged to be able to give a talk as part of the Casa Bio initiative at PAG, which is another, we have the government behind us now, which is great. And then I think the other sort of like transformative thing that's happened since 2020 is artificial intelligence. And that, I think, is one of these other factors that I think is impacting our field. So I've just sort of given some examples about the ways that some data projects are already making use of artificial intelligence. So, and also, as other people have mentioned, that, we've gone from this sort of like waves and waves of data to like, it's going to be a lot, even more so. So we really have to find more effective ways of being able to handle and manage all of this data. Otherwise, we're just not going to be able to keep up. And this, to me, also as a biocurator says, is really justification for the importance of the work that we do. So I wanted to sort of put us in the mode of like, we now have this sort of forward-thinking timeline where we are now in our last year of our 3-year grant. And as people have mentioned, we are anticipating being able to get some funding to be able to keep us going for a couple of years. But part of that going forward is thinking about what will we do next? What is the next grant that we're going to be pursuing, or grants that we're going to be pursuing? So we're sort of in this place where we've been consolidating our ideas and prioritizing things so that we can formulate a plan for the future. So we've, again, for those of you who are in Chicago, last year, there were a lot of different ideas that people put forward, and we've sort of consolidated them into various, sort of structured them along with our 4 initial goals, which were, I've sort of tried to summarize here, but I think there's just sort of a lot more. But there's a lot of ideas in the sort of data and metadata standards and tools kind of space. The need for not just the data standards and the recommendations about metadata and the standards, which are really important, but also a lot of ideas that people had about like, how do we facilitate the adoption? So I think we've heard as a theme about, we may have different standards, but not everybody is using it. And there's also the problems with the barriers towards data verification. So there were a lot of ideas about having, data and metadata validators, facile tools to promote submission, file format checkers, the importance and application of machine learning and AI to improve our literature curation and other applications of AI in software development. People had ideas about bioentity checkers and validators. So those of us who work in databases know that nomenclature is a huge problem. We just heard about this problem of like, people say they submit data, but they don't necessarily, there's no sort of validation for that. So the need for things like this and other ideas about, sort of FAIR from the start and like helping people with their initial FAIR data collection. And there were some ideas about how we could achieve, the development of those tools, including mini grants, hackathons, and competitions. Sort of, again, back into the data metadata space, there were ideas about other working groups to develop and work on these data standards and recommendations. And some of those working groups actually already have started. So things like the single cell RNA-seq data and other aspects of the phenotype data and metadata management. And then, of course, thinking about, what are some of the things that we can do to incentivize the data and metadata submission. So switching to the sort of other goals, we had also ideas about sustainability and outreach and education, or these other missions. So specifically, there are ideas about sustainability for the consortium itself. So how are we able to keep this group of people going? There were ideas about being able to, provide a little financial support and other sorts of incentives, as well as sort of capacity building for the leadership of the consortium. Along with the sustainability of the consortium, there are ideas floated about sustainability for the community databases, including, the sort of quantifying the value proposition to collaborate on synergistic projects, to do more about the data reuse and submission education. I mean, a lot of our ideas have sort of, they overlap in different sort of domains. Ideas about capacity building for staffing and potentially this notion of service centers. And then in the education and outreach space, what can we do about increasing our interaction with industry, which is one area that we really hadn't pursued. People had ideas about having more consulting and like a help desk for researchers. So like, dial a curator. We wanted to, as John was mentioning, do some capacity building and data stewardship aimed at early career researchers, folks from, HBCUs and minority serving institutions, and as well as maybe focusing some of our education and training towards reviewers, as well as our own database personnel. And then, of course, sort of, expanding our outreach and being more present within the community. So there's a lot of ideas and a lot of stuff to ingest. And I guess, my framing for this, section is, are there other things that we have missed? Are there other communities or individuals or ways that we can go out and seek, additional sort of feedback and make sure that what we're doing is the most effective use of our time? Who else should we be involving? I think somebody, Peter, made a comment about, interacting with FADER. And then, of course, how do we, prioritize these ideas and then identify people who are not just interested in, but committed to doing the work, because all of this is going to take a commitment by people to make this happen. And so we need folks who are ready to raise their hands and say, yes, I want to, take charge and lead this. So that's, I hope that's enough sort of fodder for thought to begin to have this discussion. So does anybody want to kind of raise a hand and sort of throw out some ideas?
First of all, thanks for that, Lenore. I'm going to just chime in because I think I'm the host for this discussion session. So appreciate your wrap up here and thoughts. Yeah, and so Lenore brought up a bunch of different ideas, things that we've talked about in the past, and different scopes of progress where we might think about going towards in the future, sort of from, a future working group level to thinking about grants, beyond the scope of the RCN. We have an RCN right now. We have the opportunity to extend it for 2 years, but an RCN pays only for, this network to continue. And so for bigger things to happen, software development, things that cost more money besides a network and a coordinator, essentially, what should we be thinking about, and how do we do that? So, please, let's open the floor and feel free to either raise your hand and say things directly or add things to the chat. And we have Sarah, I believe, taking notes for us in a notes document so we can capture all of your feedback. Appreciate that. Jacqueline, go ahead.
Hi, this is awesome to see like all the new ideas. I particularly have been really interested in myself in using more AI to just help me curate data. And it's something that I'm finding a little bit hard to just to grasp around because I don't have the computer science background that I need. But like it would be awesome if we could. I know this isn't an issue. The issue for everybody is that just maybe as a group, either a working group or some sort of maybe consultation with other uh, people who have done this, because I'm noticing a lot of machine learning biocreation going on in the human space for NIH, and I think that if they're doing it for human, there's no reason why we can't do it for plants and animals. So I just feel like we need to do something for that because it would really help. That was just my 2 cents.
So working group on AI to facilitate biocuration. Thanks, Jacqueline. Rex has his hand up. Go for it.
Yeah, I was thinking in the terms of hackathons and how we can be more inclusive. If we do have a hackathon, could it be on the campus of an HCBU?
Yeah, good point. Thanks, Rex. All right, Sushma, you're up next.
Yeah, so we have been trying some AI to do some of the gene curation, and so initially the Reactome project used ChatGPT, and they they did curation. They also compared how good it is with respect to manual curation, so they kept the timing and everything. So one of the thing with the journal software like ChatGPT, it has a lot of hallucination problem. And the way it works is the semantic structure, right, of the sentences, and the information that it brings is not very efficient and not very accurate. Lately at OSU, we had AI week, and I have actually gone to some of the workshops that were offered by Microsoft and also NVIDIA. And they have a couple of new tools that are more accurate, and they are actually mining information from Grameen ensemble database. They are connecting to UniProt. They are, connecting to the abstracts and the PubMed that are available. And I think they have some utility, and, I'm very excited to use that and, share some of that. So I have checked so far close to like 100 genes that I have well curated with my students by reading hundreds of papers, and then we got some of the AI generated and compared those. So we have some good points. We can't 100% rely on those as of yet, but there is a use that we can enhance and increase and become efficient using the AI tools.
Okay. I have a quick follow-up question that's sort of egoistic of me because I'm curious. I'm going to butt in line. So Sushma, you mentioned Grameen, et cetera, mining data. So how, or the researchers mining data from databases, how AI ready are our AgBioData databases? Where Jacqueline brought up, curators need to use AI to curate the data, but then how are we doing as databases in terms of making our data web service accessible and structured in the way that somebody could pull data from Grameen and Tear and MazeGDB and Soybase and easily create their study for machine learning. Do you have a sense from what you mentioned then how we're doing there?
So I think one of the thing about how do you mine data using AI is that you have to learn a little bit what are the better prompt. And so I think how do you define the prompts and you have a good prompt, and then you ask the question. And good thing with, for example, Copilot from MSN is that it will generate the following 3-4 questions. And so, for example, if I say, provide me a summary of, say, lazy one gene, it gives me summary and publication or something. And if I say, provide me, summary of the 80 whatever gene, then it brings the information from the databases. So it's kind of nice that, it brings a different information when you use the popular gene name versus the standard gene ID. So it's kind of, yeah. And
Okay.
it is picking a lot of DOI IDs. I think it is getting some information from the ResearchGate. My experience is that it is mining information only from the abstracts and not from the detailed paper or text images or anything. So the detailed level of the information is missing.
Yeah, I've looked into it a little bit, and I think still in both Monica mentioned how our database is ready. So most of our data in our databases are in underlying database, right? So you have to query them. They are not all in text in our website. So I think both needs to be a little bit advanced. So our databases need to be more textual, for current AI to work, because it wouldn't go into the database and do the query, right? So and also AI, I feel needs to be more advanced in this to do this type of jobs, so that it can actually go into the database and query. But for now, I think the commonly available AI is just looking at the text. So it can look at the PDF or whatever on the website, but then it wouldn't go into the database and then do the queries.
So, yeah, so there is also the AI that is looking specifically for the images. And then there is AI that is specifically aimed for adding some creativity in terms of language and also figure making to everything. And then there is a third part is the generative AI, right? So I think there are different type of the AIs, and the list will grow, and I think their utilities will become more specific, some more accurate, some more imaginative and more creative. So learning about I think I like the question Monica has posed, and also Sukh, that we have to have, somehow tweak our language, and we have to, when we curate our data, we have to think about, AI prompt and structure, how they are looking into. So that will make our databases more accessible.
So 2 working groups, one for biocuration using AI, and a second for making our data more accessible and findable for AI algorithms in particular, which is part of FAIR, but still we need to make sure that that's happening. Okay, good, great discussion. Tanya's had her hand up for a while. Go ahead.
Thanks. So I wanted to add in my support for the idea of increasing our outreach at conferences. From attending and being at the AgBioData booth at HAG, I could tell you numerous anecdotes about people stopping by the booth and being like, oh, I am building a database for my particular organism. It would be really interesting to learn from what you've done. Or I work in this particular plant, but I use these others. And oh, now I realize that all of these other databases exist. So as far as like raising awareness for the individual databases, but also for the work that the AgBioData community does as a whole, to kind of like, you have a new database. Hey, there have been so many that have done this before. We can teach you this data type. I'm integrating this new data type. Or my country is setting up a repository for this particular data type, and we need it to connect with others. You're people I can talk to. And so HAG is a great place for that. But I suspect that there are other conferences where we could reach additional people. And so largely we've had plant databases that we're interacting with. I think we could take the opportunity to go to where the animal databases are, to expand in that area and bring those people in. And that adds to our sustainability as an organization, because we're bringing in new people. Maybe we're bringing in people who would potentially be involved in leadership. So aside from working group, just kind of making our presence stronger and broader. I would like to advocate for that.
Yeah, it's a great idea, Tanya. Just to follow up on that, and then Sushma, I would probably be one of those people who you're talking to, since my community is not in plants, it's insects. And so the question for me would then be, what has AgBioData produced that would be of interest for the larger insect community, for example, if I went to the Entomological Society of America meeting. And so I think we've had a lot of products now that are worth shopping right now that are not plant focused, right? We have the education curriculum around fair data. We have all these other aspects. So, I mean, not to put more work on ourselves, but it sounds like to make this more accessible for other people like myself, or others who might go to livestock workshops, for example, to have, like basic slides up that we can share and provide for people to say, hey, if you're going to Entomological Society or wherever, here's some stock slides of vetted information from the steering committee and PIs on what we've produced so far and what might be of interest to a general community. And then please, provide specific examples that would be useful for insect or livestock or whatever. Is that sort of what you were thinking about? Or
yeah. And I think we're in a really good place now, because the history of the RCN is there are a number of white papers, there are a number of educational materials that are ready to be shared, or are really close to being ready to be shared. And so it's not just this idea of, oh, we're building this consortium. It's like, no, this consortium exists. Here are resources we have. You can use them, come join us. But we need to go to where these people are.
Yeah.
Or to go to more places where they are.
Thanks, Tanya. Sushma.
So I think one place we should go more, and where we have very little representation from the plants and also from the members of the AgBioData, is the Biocuration Conference that is organized by International Society of Biocuration. And they have a lot of things that we are working on humans and model organisms and everything about software tools to data standards to use of AI. And so we have a lot of opportunity to learn there. And I think the representation of AgBioData is not there. I was in that conference in the March. I think I was the only person from the plant, from here, and apart from some people so that is one group. The other group I have also asked Ilenka and a couple of other people is the NASA analysis working group that has a lot of Gene Lab open data from the NASA Gene Lab. And I have been part of it, and some of our folks also from Plant Reactome Group are part of that. So I gave actually the genotype phenotype white paper talk there, and they are working also on open data. We call it FAIR, they call it open data. And NASA has the TOPS training for the open data, a series of things. And now they require every PI who submits the data at least to have that TOPS training, which is similar to FAIR training. And they're working on the similar issues, the standardization of the transcriptomic and genome sequencing data, phenotype data, how to go forward, and all kind of things. So I think these are two places where data curators can learn a lot.
Yeah, great suggestions. Thanks. Lenore.
Oh, yeah, no, I was just thinking, but with ISB, I think the last couple of meetings have been places that are very expensive for folks to get to. So, I think Jackie said that she's planning on going when it's in Kansas. So that might be a little bit more accessible to most of the people here, and I'm already hearing like a number of, sort of synergies and collaborations and more connections that we can be making. I know I've been a member of ISB for about 1,000 years, but I don't think I've been to one of the meetings since it was at Stanford because it's just money. Yeah.
Can we provide any of our members or, steering committee members or working group leaders some mechanism or write a grant maybe from NSF or USDA to support participation? So for the next upcoming meeting, maybe that could be an idea.
Yes, we are strongly interested in the RCN extension to provide funds for sort of AgBioData ambassadors to reach out to the larger community on the work that we've been doing and generate more interest and support our community that way to be able to facilitate that. So yeah, great point, Sushma, and we want to facilitate that. So Diane, we will need to talk to you about that mechanistically in the future. So yeah, great points. How are we doing on time, Anarita?
I think we started 10 minutes late, like 5-10 minutes late. So I think we have another 10-15 minutes to go.
Okay, good, thanks. Okay, so I'm looking at the questions here. So we've had a lot of discussion on sort of AI, what ideas or what future work we might want to do in a working group format on those levels, and education and outreach. Maybe I'd want to ask a bit more on the level of who and where. So who else should be involved was one of the questions that Lenore posed, and we had some ideas here about ISB doing hackathons on campuses of HBCUs, et cetera. Are there, so the question was who else, but I might also want to pose this as how else. So, if we don't have enough synergy right now with ISB, the International Society for Biocuration, while we have a lot of biocurators here, why is that? Is that mainly a cost thing, or are there other, points of synergy that we could be exploring more with those communities that are related? And ISB is just an example. If there's other communities that you're thinking about, yeah, that's good too.
Yeah, I think there is another one, data reuse community, that is Pacific Symposium on Biocomputing. So that is another group also looking heavily on the data reuse, and I received the award earlier this year for data reuse from them. So they have this Data Parasite Senior and Junior Award, and they have every year annual conference. So that could be other group that we could join.
Okay. What was the data reuse from what organization? Biocomputing?
It is called Pacific Symposium for Biocomputing. And they
Gotcha.
have a small society and group of people, not plant people there. So I think I was the first one from plants there, but a lot of microbe and human and other model organisms.
Yeah, no, great suggestion. Chat from Janitor, she was thinking of the NIH data science community. Jennifer, do you want to elaborate on that a bit?
Yeah, NIH sends an email out, like, I think bi-weekly, based on their data science office, and it routinely has presentations on data reuse, data management in different communities. Some of the recent ones were around using AI in biomedicine, how to make your databases, more AI compatible. Right. So I think a lot of topics that they're discussing, could be relevant to this community. It's definitely biomedical. So we have to do a little transfer learning, but I think some of the stuff they do talk about could be useful for us.
Yeah, that's a great point. There's Layla has a comment. There's a lot of overlapping interests and synergy with advocacy efforts with the groups that attend the metascience group. So yeah, thank you for that. I'm assuming that's a library focused group, Layla, or is that or
it's not
information
actually.
science?
It's focused on just improving the practices in science. So they're very interested in fair data and developing and advocating for standards and shared infrastructure.
Okay, another great idea. Tanya brings up RDA, very relevant. That's more sort of like a parent group for us. And there, the conferences around the world. I think David Molick, actually, of AgBioData, is also active in RDA. I'm hearing a lot of this, and I'm wondering too, so there was the comment from Soshma before in terms of, funding. And I've been personally interested in RDA, and I've just never found the time to actually, go to one of their meetings in addition to that. So I do wonder, if we push having, funding for people to represent AgBioData specifically at another community would be useful, just to incentivize, the time. And if you already have a foot in somewhere else, or we're thinking about putting your foot into another community, acknowledging the time that it is that there are some financial resources and maybe a template to get you started to advertise AgBioData and identify what those synergies are. So perhaps we should do that. Ag groups and RD that overlap in our missions. Lenore, you had your hand up. Sorry. Yep, all there. Yes, RDA meetings they're virtual. Okay. Makes it easier. So then it's just the question of finding the time, which is always hard. And the incentive.
There's the time difference because they're often in Europe.
Oh, yeah. Okay. Awesome. Okay, we probably have another 5 minutes or so left, but Anarita, please cut us off if we're running over. So the last question that Lenore had was how to prioritize ideas, and then who is interested in committing to doing the work. So we just talked about time, and doing this sort of work within AgBioData also takes time. But there's, I think, we're probably at a point now where we've generated a lot of ideas, and it might also be, fertile ground for people to think about, what have we produced and ideas have we produced that you could write grants about also, right? So I'm hopeful that, and I think everyone here is hopeful that the discussions that we've been fostering here will help, you all when you write grants to incentivize that and help you move further along there. But yeah, how to prioritize ideas and who is interested in committing to doing the work. So getting, fresh excitement for the group. Are any thoughts about that? John has a comment and Anarita, so help us identifying calls for grants. So yeah, so thinking creatively about funding and where else to apply. And then John had a comment on we need to structure our work, I guess, as more structure for the work I'm reading. So management and for member databases would be helpful. Do you want to elaborate on that, John, or?
I just think we did, for the forthcoming couple of years, we need to make these action items. We've had a lot of discussion and we'll have some new discussion, but what can we, okay, now we've got ideas that we can act on. So, make it focused on, okay, we're going to find grants amenable to this task, right? We're going to meet with specific institutes on what they need, that kind of thing.
Yeah.
Yeah, and I think, John, that would help also in identifying individuals who are best placed to actually do those, right? Have the right expertise, and then hopefully have the bandwidth or the lack of ability to say no, that then would be best placed to move those things forward, right? So it depends on, are you thinking, oh, we have to do outreach to other groups, which of course then people from those groups would probably be best placed, but then also like if you're going to move into the AI space, then okay, who has the requisite knowledge, right, to move that forward? Because yeah, then at least you're presenting somebody with here's kind of what we're trying to achieve, and so they can then to themselves ask themselves like, okay, is that something I could have the bandwidth to do, right? Does that seem like it fits into my schedule, right? Just gives a little more structure around the ask.
Lenore.
Oh yeah, I was just going to say, like, one of the nice things about all this work that we've done is that, we've really done all the hard work to sort of identify what the needs are and the justifications for things, right? We clearly keep coming back on the need, the certain software kinds of things, the validators and, the educational resources and stuff like that. So, I think there's a lot to be sort of picked through and threads to be pulled in this that I think really are potentially very impactful. So
yeah,
I know we're all really sort of super busy and nobody likes writing grants.
Yeah, that's a great point. I mean, we've done a lot of, there's been a lot of survey work to initially in the first year or two, which thank you to everyone for responding to those, but where we were initially trying to figure out what the needs are. And so there's papers written that do describe those. So that's a good point. That's that we should turn, to the Data Federation on Ontologies white paper, or the genotype to phenotype, or the ones that are coming out now to help us put those together. And I noticed some themes during the workshop talks in the past couple of days. I heard metadata multiple times every single day. That's still a thing. So yeah, I think, yeah, we've done a lot of work. And so the writing committees would have a lot to draw from to help define those priorities. Okay, just monitoring the chat real quick. Sunita has some great stories in the chat that you can look at. Random generation of PubMed ideas. That sounds great. So yes, grain of salt there, a couple of grains of salt. Okay. And
yeah, we call this hallucinating effect.
Yes.
Completely
Auto-generating PubMed IDs.
Yeah.
It's great.
Yeah, completely fabrication.
Yes. Thought about lowering the barrier for data submission without losing quality. Yeah, that's been a theme. Enforcement of data submission and DMP adherence. Yes, those are good themes for us to think about. Okay, so I think we're at quarter till. Any last burning thoughts from the group?
Thank you. Thank you to everyone. This takes a lot of work.
Yes, thank you all for participating. So I guess we're wrapping up now. So thanks everyone for a wonderful 3 days in discussion. As Lenore said, thanks to Anarita and everyone for putting this on, and yeah, for being part of our community. So it's been fun. And yeah, see you all online.
Thank you, everyone. Bye.
Bye.
Thank you. Bye.